Food chains often tell us about the feeding relationships within communities. The first organism in a food chain is always a producer. These are plants and algae, and they always start the food chain as these organisms produce their own food using light energy. The sun is the source of energy for almost every food chain. Primary consumers are animals which eat producers, so on land these would be herbivores. Primary consumers are then eaten by secondary consumers, and these consumers can then be eaten by larger carnivores referred to as tertiary consumers. This could be one food chain: grass as a producer, grasshoppers as primary consumers, rats as secondary consumers, and then snakes as tertiary consumers. Primary consumers have different issues to secondary and tertiary consumers. Predators can be secondary consumers and above, while prey animals tend to be primary consumers, but they can also be consumers further along the chain too. Primary consumers eat plant material, which means it is easy to find and doesn't need to be caught. However, predators need to catch their prey before eating it. Plant material does contain cellulose, which is difficult to digest, while meat is only protein and can be easily digested. Predator and prey numbers can vary in a food chain. When there is surplus of food, prey number increases. This leads to an increase in predator numbers. However, this eventually means that the number of prey will decrease again, due to them being eaten by the predators, which in turn leads to less predators, and this means prey numbers again will increase. This cycle takes place in stable communities. Communities can be studied in fieldwork. In fieldwork, quadrats are used to see how the biotic and abiotic factors affect the distribution and abundance of organisms. Quadrats are simple and effective ways of estimating the population size of plants and small creatures. This is usually done in a 0.2 metre squared area. When measuring the distribution of organisms, it is important to place a quadrat randomly. So, if you did this on the school field, you could close your eyes and place a quadrat, or you could use a random number generator with coordinates. You must also make sure you repeat this many times to get a good understanding of the distribution and make the results valid. Using your results, certain calculations can be done. A mean number of plants per quadrat can be calculated by adding all the readings together and dividing by the number of readings done. The median is the middle value of the data, the mode is the most frequent reading, and the range is the maximum value minus the minimum value. These calculations can be useful in your analysis. Transect sampling is another method, and you're probably most familiar with a line transect. A transect is where you sample along a set line and measure the distribution of organisms. Transects are useful when the conditions change along it. For instance, a transect could be done from a shaded area of the field to a non-shaded area. Along this set line, you will sample at set intervals, for instance, every 2 metres. The next required practical is to do with this topic. The practical is about measuring the population sizes of common species using random quadrat sampling and transect sampling. Using the random method, the population size of a species is estimated by doing the total area divided by the area sampled times the number of individuals counted in that particular species. Using the transect method, you could measure a varying factor as you move along the transect, such as the pH of the soil at each interval.